Welcome to our lesson number 5, and today we are going to study the qualitative analysis, the importance of qualitative analysis, different kind of qualitative analysis that we do in pharmaceutical analysis. We will also study the crucial aspects of this method, and let us start the video. Lesson number 5, quantitative analysis in pharmaceutical chemistry. So dear students, today we are going to study this chapter. So qualitative analysis and quantitative analysis are the two main aspects of pharmaceutical analysis, as I have already told. On one hand, when quantitative analysis tells us about the amount of a substance in the sample, on the other hand, qualitative analysis will tell us whether something is present or absent. So we are just identifying the components in the sample. We are not concerned about the quantity of that substance. Now in pharmaceutical chemistry, again, likewise, qualitative and quantitative analysis was important. In the same way, qualitative analysis is also crucial. It's very important, because sometimes you just want to know the drug substance. You want to check whether some impurities is present or not. You just want to check whether certain kind of excipient is there or not. So the principles of qualitative analysis, they focus on just identifying substance. You are just checking whether something is present or absent in the sample. So for that we utilize a variety of techniques. Okay, and these techniques are just focused on the characterization, or you just want to know the chemical and physical properties of that substance. You check those for those quality, you check for those things, and you check that substance is present. Now, identification of pharmaceutical compounds. Why we are doing identification? Why qualitative analysis is on the emphasis? So it says that it's very essential to ensure the authenticity of the pharmaceutical product. Sometimes we just want to know whether our tablet contains the specific drug or not. Like for example, if we are having a paracetamol tablet, and say we just want to know whether paracetamol is present in order to identify that this is the correct tablet, so we just do the qualitative analysis. We just want to confirm the presence of our intended active pharmaceutical ingredient, or on the other side, we just want to detect whether some potential impurity is present or not. For both the thing, we just want to know whether something is present or not. So for that we do for qualitative analysis. Now in qualitative analysis, a very important technique is used, which is known as spectroscopy technique. Spectroscopy, in the previous videos, in the previous lessons, I have already discussed about that. There is a UV visible spectroscopy, there is infrared, and there is NMR. In UV visible spectroscopy, we are using ultraviolet light or visible light, and these ultraviolet light, UV light or visible light, is subjected on other sample. The sample absorbs some amount of ultraviolet light, and rest of the amount is transmitted. By the amount of absorbed light, you see that what is the lambda, what is the wavelength of the absorbed light, and by that wavelength you easily identify the type of double bond, or you identify the particular kind of structural pattern. Now in infrared spectroscopy, what we are doing, infrared spectroscopy is basically used to check the type of bond, whether it is C N bond, single bond, C double bond, C double bond, C triple bond, C bond. So that kind of bond, the type of bond, if we want to identify the type of bond, we are using infrared spectroscopy. These are all just qualitative analysis. We just want to know whether the particular kind of bond is present in or not, and by this identification we easily identify the characteristics of that sample. Now the nuclear magnetic resonance spectroscopy, it basically tells us about the structural information of the molecule. And what kind of structural information? It gives us the number and types of proton. Basically we are dealing, most of the time we deal with the number and type of proton. For example, in ethanol CH3 CH2OH, we are having three kinds of proton. 3 protons are of the methyl type 1, 2 protons are of the methylene group type 2, and 1 proton is OH group. So the number and types of proton, these are identified by nuclear magnetic resonance spectroscopy. Now there are other techniques of analysis, qualitative analysis as well. One major important technique is chromatographic technique. In chromatographic technique, there is a kind of chromatography known as thin layer chromatography and gas chromatography. They can be used to separate the components of a sample and identify the components of the sample. There is a factor called RF value. Dear students, you must know it. RF value stands for retention factor or retardation factor of the sample. By the help of retention factor RF value, you can identify or you can perform the qualitative analysis of the sample. Now there are colorimetric tests also. Colorimetric tests are done, to colorimetric means you are basically focused on color. You are seeing the color and you are identifying the special functional groups present. Now what is the importance of qualitative analysis in drug development? Number one, it is the early stage identification. You can identify the drug molecule. You can also characterize the impurities and byproducts which are present in the drug molecule, and that's why it helps in drug development. Now the examples of qualitative analysis in pharmaceuticals. See, we confirm the identity of an unknown tablet or capsule, and you can use the IR spectroscopy. Say for example, there is a tablet of paracetamol. Again, I'm using the same thing, paracetamol. So paracetamol, it is para amino phenol derivative. This is para amino phenol derivatives. This is NH, C double bond O, single bond CH3. Now we in this tablet we want to know whether paracetamol is present or not. So what we are going to do is we will subject it to infrared spectroscopy. Infrared spectroscopy will tell us, okay, there is a phenolic OH bond, there is also an NH bond present, there is also C double bond O carbonyl group, and there is also CH3 group. It will tell us the type of bonds present there, and by this information we can easily tell that, okay, infrared spectroscopy is telling us that the type of bonds that paracetamol is having is present in the sample. So by that way we understand that PCM is present. Also another example, say we want to identify PCM in more in another way, or we want to be more confirmed or assure that this is paracetamol. So one thing we did is that we did IR spectroscopy. Another thing we can do, we can do its mass spectrometry. Mass spectrometry, by when we will do the mass spectrometry, it will tell us the molecular weight of paracetamol. If the molecular weight comes out to be equal to that of PCM, then we can say that PCM is present in the tablet. So by that way we can use qualitative analysis in the pharmaceuticals. Now we also detect impurities in the drug sample through TLC. TLC stands for thin layer chromatography. So thin layer chromatography is nothing but there is a glass plate in which or the aluminum plate in which silica is poured. In that plate we are having our sample spots. We are having the spot of our component, our sample, and the sample, if it say contains three component A, B, C, and when we will run the mobile phase, A, B and C, A, B and C will be separated and they will appear as a spot when we will perform visualization, and we can easily identify, okay, this is A, this is B and this is C by understanding, by recording their RF values. RF value will tell us ki C is what kind of molecule. It will also identify A and it will also identify B. So by that way we can identify A, B and C, and if A, B and C are impurities then it will be known to us. Now another thing that we can do, do with the qualitative analysis is we can verify the presence of specific functional groups in the compound by using colorimetric tests. So these tests will identify the different kind of functional groups that are present. Now summary of this chapter. Now point number 1, qualitative analysis, it mainly focus on identifying the compounds, the components which are present in the sample. We just want to check whether something is present or absent. We don't want to focus on the quantity because quantity is focused by quantitative analysis. Second thing is, there are the principles of qualitative analysis. They are focused on characterizing chemical and physical property without quantification. See, you can easily identify something. If you want to identify something in your life, even it's a very broader concept, if you want to identify something, then you have to check for certain chemical property or certain physical properties of the thing. If you can check those physical and chemical property, you can easily identify that substance. Like for example, in UV, we are basically checking the electronic excitation, because when we subject UV to a substance, there is electronic excitation. By mass, we check the molecular weight. Molecular weight is also the property. Number and types of proton, this is also a chemical property. We identified by NMR. So these are all physical and chemical properties which are under the focus of the analyst, and analyst identify these physical and chemical properties, and analyst understand what kind of substance is present. Okay, now the spectroscopic techniques. There are spectroscopic techniques, or there are chromatographic techniques, and there are colorimetric tests. They are very, very important. They are playing key roles in qualitative analysis. These techniques are extremely important, and we frequently use these techniques in modern days to identify some substance. Now the last thing is that it's very, very essential to ensure the integrity and identity of the pharmaceutical compound. We just want to know whether the something is present or not. It will just check it will confirm the identity, and that is why we are performing quantitative analysis, qualitative analysis. Thank you so much. So dear students, I hope you have understood what qualitative analysis is, and you have also studied the different spectroscopic analysis. Now you have clearly understood that qualitative analysis is used for the identification of compounds. Ideally, we are also having a separate chapter for the spectroscopic techniques. Now let us move on to our next chapter, which is chromatographic techniques. In the next video we will study that, and till then you keep practicing, keep revising it, and we will meet you in the next video. Thank you.